Hello, welcome all for the HoliSoils Webinar number 8. Today we will talk about long-term nitrogen fertilization and how that enhance tree growth and soil carbon retention in boreal forests. We start with the presentation by Anni. The title is Understanding the Tree Growth Mechanisms Behind Nitrogen Fertilization. Anni will talk about 20 minutes, and after that we have Boris Tupek, and he will talk about the reduced microbial respiration sensitivity to soil moisture following long-term nitrogen fertilization may enhance soil carbon retention in boreal Scots pine forest. And at the end of the session, we will have 20 minutes for questions and replies. And in Teams, the chat functionality, in Zoom, the chat functionality is not operational right now. So when you have questions, there is a Q&A button in the bottom. So please add your questions there and we will take them at the end of the session. And now we could start. Anni, please go ahead.
Thank you, Aleksi. Let me start by switching the slide. Yes, here we go. So, my name is Anni Palvi. So, I'm a doctoral researcher at the Finnish Natural Resources Institute, which is Luke among friends. And Aleksi is actually my supervisor. So, I'm very thankful that he's here now and have time to be our host because he's been following my path, of course, in Luke, and very well knows also this study that I'm talking about today. So I'm talking now about Scots pine and trying to understand how the nitrogen fertilization works in tree level. And here's the paper that my talk will be purely based on. So this paper was published this year in May in tree physiology paper. So it's also my topic today to talk about tree physiology. And as the title says, repeated nitrogen fertilization enhances Scots pine growth and carbon uptake without persistent long-term effects in boreal forest. So the effect of nitrogen fertilization itself is strong, but unfortunately very short-lived, and we didn't find any long-term effects in tree level. But on the other hand, we also find that there was no signs of nitrogen saturation, which means that sites still continued to respond to nitrogen fertilization decade after decade when the nitrogen was applied. So that said, I will focus more on short-term mechanism in this talk and what happens in the tree level after one fertilization effect. And we can also consider this talk like as an introduction to the soil level talk that Boris will do after me. So first I will introduce our study sites, which is Karstula in the middle Finland. It's a nitrogen-poor dry mineral soil site with Scots pine and a block design. And there's 3 blocks, like you can see here. And there's multiple fertilization experiments, but we only focus on these nitrogen plots and their control that didn't have any fertilization. And I think that it's pretty unique site as it was established in 58 already and then fertilized every decade since 1960s. So here is the fertilization that was applied every decade. And so it makes 7 fertilization rounds by today already. And what we did was tree ring sampling, and that was done in 2022. And here's a picture, so you can see an example how that kind of sample looks. And these tree rings are, so to say, like nature's own data center or data warehouse. So using that data, we are able to go back in time to produce this timeline. For example, now here we can see the basal area increment. So in the upper figure, we can see separate timelines for nitrogen fertilization and the control plot. And in x-axis, we have, we have year here, and this dashed line marks the fertilization that was applied. And what this reveals is like each decade after fertilization, the basal area increment goes up and down, back to the control level at the end of each decade. So up, down, and then we calculated this so-called treatment effect. It is like it's calculated by, it's like a difference between nitrogen and control. So in order to get rid of this so-called background noise, what is the control basically? And we can see that the outcome is very similar. So up and down pattern goes on. And next we analyzed the stable carbon isotopic composition in the tree rings, which is this delta 13C. And this value can be very useful when we try to understand the tree physiology and those processes in the trees. And in this case, we are interested of what happens after the fertilization event, but a little bit of theory behind this value is that the plants prefer to assimilate the lighter carbon isotope, carbon 12, and discriminate this carbon 13. And I must say that the theory behind this value is quite complicated, but I wanted to cut the long story short because I don't have more than some period of time to explain this. So I will simply say that this delta 13C can be described as the ratio between net photosynthetic rate and stomatal conductance. And we can think that these both values are basically related to the amount of carbon that enters the tree from its needles, and then it's reflected to the tree trunk. And here we have 3 trunk samples that we analyzed. What we see in this figure is that the lines go up and down quite randomly. So they are mostly reflecting environmental conditions. And it's not as clear as basal area increment maybe. Like in theory, for example, the peaks are probably cause of high temperature or dry year, or maybe there was a lot of sun, so there was no clouds, so solar radiation might be high. For example, here probably it was dry autumn, but we can also only like speculate. But in this study, we are not really interested of the weather itself, but more like tree physiology. And so we can presume that these both plots, control and nitrogen, they have the same environmental conditions. So again, we calculated the difference between 2, and we can get this treatment effect once again. So maybe we can see here some pattern, or do we really, like, is it that we want to see some pattern? And yes, one thing that we did also, like we divided the samples in early wood and late wood. So early wood is this upper figure and late wood here. But anyway, it's pretty hard to say from here anything. So we investigated this a bit closer, and we ran the superposed epoch analysis for the treatment effect. So these are all treatment effects. And there's delta 13C for early wood and delta 13C for late wood, and then basal area increment. And what superposed epoch analysis is good for, it's revealing the patterns in this kind of time series where we have multiple similar events. So what it basically did was that it calculated the mean for the whole data and then compares the values of the time series to the mean and kind of overlaying these events. And by doing this, we can maybe remove the random noise from the data and just leave the treatment signal. So the x-axis here is the year after the fertilization was applied. So the 0 year is the year of fertilization, and then first year after it and second year after it, and so on. And then there's z-score, which was simply to standardize the data for this analysis. And well, what we see here is that delta 13C early wood, there's like statistically significant signal during first and second year after fertilization, but then the signal disappears quite quickly. And a little bit similar pattern with late wood, but not as significant, or there was no statistically significant signal found. And maybe we can speculate the reasons why this is like this. But what is interesting is that the effect of basal area increment is lagging 1 year compared to the delta 13C. So in the same tree rings, there is first quite fast reaction to the delta 13C that is then followed by growth reaction in the tree rings. And it is a little bit longer than this delta 13C. Well, at this time, I would ask, like, what is this delta 13C? Because according to the theory, it is just so-called proxy, and it works best when it's combined with other analyses. For example, this basal area increment, but I will go now for needle analysis for a while. So we used the needle mass of 1,000 needles and nitrogen content to analyze the needles, and this was performed in 80s. And in 80s, this was sampled like during the winter before the fertilization event, and in 83, the sampling was done during the winter 83. So this is basically, I would calculate that it's 2 growing seasons after fertilization. And what we see here that there's significant increase in needle mass and nitrogen content in the fertilization plot, while control didn't react. So fertilization increases needle mass and nitrogen content. And since nitrogen is the main component of chlorophyll, this means basically increased photosynthetic capacity of the tree. That then leads to photosynthetic rate. And now when we go back to this delta 13C and its interpretation and combine this observation of the needles, well, we can maybe make a conclusion that it is mainly this photosynthetic rate that we see in the superposed epoch analysis. So the increase we see in delta 13C is the tree rings in the tree rings, yeah, it can mainly be linked to this increase in photosynthetic rate, because there's more chlorophyll in needles. So here's my attempt to combine this information into a fluent storyline of what the tree growth mechanism is behind this nitrogen fertilization. So first, what happens after fertilization is that nitrogen fertilization increases the nitrogen availability and needle nitrogen content increases. Then we see that photosynthetic rate increases here in delta 13C. And third, there's enhanced needle growth and needle mass increases. And maybe we can also combine this, like they say, or say that it's more leaf area for photosynthesis. And then fourth, we see enhanced trunk growth. And then there's basal area increases, but the effect persists only until the enhanced set of needles is shed. So as a conclusion, the increase in delta 13C, it's very rapid, and it's gone after 2 to 3 years or so. So in the perspective, so in this perspective, this fertilization itself is actually even shorter, like the effect of fertilization, than what we see in the basal area increment. But on the other hand, the effect is sustained in the basal area increment because of the bigger needle size. So even if the photosynthetic rate goes back to the original levels, there is still more photosynthesis because the needles are now bigger in size. And then when the needles fall down, the effect of basal area increment slowly drops to the control levels like we saw in the first picture. So a little bit more conclusions. I want to argue that the duration of the fertilization effect on basal area increment is connected to the lifespan of the needles, which means that the fertilization effect slowly diminished when the needles are shed. And it is this enhanced needle biomass that enables the enhanced basal area increment. So in climate smart forestry, maybe we should be focusing more on the needles. Like usually the discussion revolves around the tree trunk biomass, but I argue that climate change mitigation and adaptation, if we want to consider those, the needles are actually even more important. And I want to finish with my own quotation. Because usually this nitrogen fertilization is done once or twice at the end of the forest cycle just to maximize the tree trunk biomass. But our research shows that the effect of nitrogen fertilization in nutrient-poor mineral soils is strong but short-lived. And for optimizing forest growth and carbon sequestration, the most effective approach would be apply small amounts of nitrogen fertilization every few years throughout the entire lifespan of the forest stand. And maybe I might continue this by adding that the use of slowly soluble nitrogen forms could ensure a more sustained nutrient supply over time. There's been discussion about this slowly soluble nitrogen, but I don't want to type it here because I haven't really studied that. But yeah, here's some things to consider. And Now I would like to thank you for your attention and to give space for Boris to go on from here. So thank you. Is Boris here?
Yes, I'm trying to set it up. I did, yeah. So thank you very much for your very nice, engaging presentation and preparing the floor for the soil for a connection between the effect of trees and effect on soil greenhouse gas emissions that I will be talking. It was excellent. So I try to now show my presentation as well. Okay. Yeah. It's not as smooth as yours, but it should do. Can you give me feedback now if you see the big slide or just presenting slide or?
Now is the presenter mode.
Okay, so okay, so it should be now.
Yes.
Okay, but it's not the first slide. Okay, all right. So my talk shows that is trying to show that there is a difference in how microbes' sensitivity to temperature and moisture is affected by added nitrogen in originally nitrogen poor boreal Scots pine forest. And this work was done also with Aleksi and also other co-authors listed here that I'm grateful. And we were funded by European Commission and under this umbrella of HoliSoils project. So it was a great opportunity for our study. And the study is basically site is the same, and probably the design that Anni you showed location is the same, but here we have like plots, study plots for soil respiration. It is D, and for example, you can see there is a fabric used to cut the tree roots, so we can see only a response from soil respiration, not so much from autotrophic root respirations from the trees, but that's what we are interested. And in this map, we see like micro topography in this site, and these green dots, if you can see, they are control, and yellow ones are nitrogen, and we have like this kind of setup replicated six times for nitrogen added and for control. And this is the forest, and we are in Finland, so we also have some snow there. But we did the measurements mostly during vegetative snow-free season, but also it included some measurements with snow early in spring. Basically, with this study, we hypothesized that nitrogen will change CO2 fluxes, methane, and nitrous oxide. And for CO2, because of already known impact on nitrogen on biomass, that it's like this is the main reason to increase biomass or tree biomass production. So we also hypothesized that this increased availability of carbon as litter fall will increase heterotrophic soil respiration, and but because the microbes will not be anymore with added nitrogen, so nitrogen limited, they would not need to mine for nitrogen so much. So the like their carbon use efficiency should be better, and they don't need to decompose so much material. So the respiration that is normalized compared to soil carbon storage will decrease. So that was like our assumptions, and we expected some changes with methane oxidation, but I have a problem with the mouse. Okay, with methane oxidation, and but we didn't know which direction it will go, because nitrogen could enhance methane oxidation microbes, but it can also compete for ammonium availability. I am not expert on this methane oxidation processes, but we expected that there will be a difference. And with increase added nitrogen, we also expected that nitrous oxide emissions could increase. So we have the same approach that we first study what is the difference in the trees, in the tree biomass and litter fall, and then we look control versus nitrogen on soil organic carbon stocks, and we studied soil greenhouse gas fluxes, temperature and moisture biweekly during seasons of years 2021 until 24. So as Anni, you showed only the time series from the start or from 80s, from the very long time series of this biomass tree ring measurements. We don't have the greenhouse gases for all the period. We only studied greenhouse gases for the the period in the last decade after last fertilization, and so we try to look on these fluxes, what are statistical differences, but also a little bit what is the process differences, and this was expressed by dependency of respiration on temperature and moisture. And we could look at this dependency with just polynomial function, this lowest, that could inform us approximately how it could be, or with more informed mathematical equation as shown before, like under these equations what we can see. And as expected in the time series of biomass, we can see increase in the biomass with nitrogen fertilization. That's figure on the left, and then in the middle increase with litter fall. This is like measured litter fall from those components like needles, twigs, and cones, and we can see some increase with soil carbon stock, but you can see that the error of these measurements is pretty large, so we are not sure if really soil carbon stock so significantly increased. When looking on CO2 respiration, heterotrophic respiration, and methane and nitrous oxide, then we found that yes, as expected, respiration of the fertilized plots increased compared to control. But yeah, we started to think that okay, this is like comparing apples and pears, that we should really look what is the respiration effect in comparison to stock, to soil carbon stock. So when we normalize respiration by soil carbon stock, those measurements, and that these are a little bit different units, so they are like gram of respiration per microgram of carbon of respired per gram of soil carbon stock. then we can see no significant difference. And there was a little bit of reduction in nitrous oxide, in methane, but these are like very small units, extremely small, but we can see some significant changes. So, because these measurements here on CO2 are measured bi-weekly, and we have now a big cloud of points, and okay, the mean values are maybe different, but as Anni, you showed that we are looking on the time period that is like several decades, and changes on annual levels as well. And these values, they might not represent well the annual values, the annual balances of CO2, for example. So for those purposes, we are using those mathematical functions with dependencies on temperature and moisture with continuous data to upscale to annual level. And when evaluating these dependencies that are used for calculation of annual budgets, we found that there are differences between control and nitrogen fertilized plots in response between this normalized respiration to temperature and normalized respiration to soil water content. And don't worry so much about this ratio. This is so we can see these responses together. So they are basically affected interactively. At the same time, respiration is dependent on temperature, but also on the soil water content. But this is the way how we could see these responses when you separate them. And yeah, this for temperature they are a little bit similar, but we have differences between this LOES, this polynomial fit, and our mathematical model. So we could say that perhaps the temperature sensitivity increased, but it's not so clear. But what is really striking here, and it agrees between the polynomial fit and mathematical model, that we have much more pronounced dependency on moisture for control plots, but not so much for nitrogen. And this we could interpret for nitrogen added. And this we could interpret that if the microbes are not so much nitrogen limited, then they are more tolerant to the moisture changes. And when we apply it to the model and simulate the annual values, first daily values here, this time series, and here are the weekly measurements and their error bars. Like first year was it was pretty different to other 2 years. There was like a drought period, and we could see that these nitrogen plots, they have, even though they show lower sensitivity to moisture, they still show some dependence on moisture, and both black and orange line decrease during this drought period, and they both increase during rewetting. And yeah, we had these are always the problems with this kind of models, that it's very difficult to model rewetting effect. And here we can see that yeah, probably it wasn't modeled so well, because in both cases the measurements show that when rewetting the respiration increased. So if these like extreme values, then they show like similar responses. But in general, overall, this control line. When you sum it up, there is a little bit larger. The measurements are a little bit larger than the simulated values than those that are from nitrogen added plots. And on annual level, when you compare it, what is the percentage of storage of soil carbon storage, then these nitrogen fertilized plots overall had lower, like around like average of these three will be around 10, while this one is around 12. So we could conclude that the control plots annually respire more compared to their stock, soil carbon stock, than nitrogen added plots. So basically, this could be our conclusion that we could see this positive reduced emissions of carbon dioxide, but I didn't explain so much, but in the one figure we have seen that there is slight reduction of methane uptake and increase of nitrous oxide emissions, but these were really low compared to in magnitude compared to changes of carbon dioxide. And another conclusion I think is pretty strong evidence that soil moisture should be considered when we do this annual upscaling, and when we consider soil moisture impact, we could even conclude that annually there is reduced soil carbon loss that could lead to increased soil carbon retention. And basically combining this knowledge with climate change mitigation potential, if we can say that we can have a reduction in greenhouse gas emissions, then this has a potential for climate change mitigation. And I think that we think that this nicely showed how the sensitivity of microbes to temperature with changes in warming temperatures in the climate and changing moisture could have impact on overall in addition to biomass changes of biomass in the trees. So thank you very much. This study was also published and is available in Biogeosciences. So now I will give the floor back.
Okay. I hope you can all hear me. Thank you, Anni and Boris. Very nice presentations. And we have a couple of questions. Maybe we could start with Boris. This is a question by Andis Landis from Latvia, and it relates to the trenching. So how long after root cutting measurements of heterotrophic respiration were started?
It's a nice question.
Less than 2 years roots. Yeah, and then he continues. There is this risk that there is a overestimation of CO2 flux. So Boris, please.
Yeah, thank you very much for the question, and I think it's a good to consider definitely the time period after the measurements started, and we started measuring pretty soon. Yeah, so we can see in respiration some of the impact of tree roots decomposition. So there is like additional carbon available for microbes from the beginning. So it's not just the organic matter from the humus, but I think the method was the same between control and nitrogen. So if there was impact of the trenching, then it should be the same in both, and we could see the signal changes. So I think maybe the rates of decomposition initially were larger, but otherwise so it could have impacted the sensitivities to temperature and moisture, but probably the same way in fertilized and not fertilized. Aleksi, I cannot hear you.
Oh, sorry, I was muted. Maybe I will take. There is another one from Andis to Boris, but in between we take one for the Anni, and this is more policy related. So there's a question about actionable steps for policymakers, scientists, and practitioners based on the research. So what, Anni, based on your results, what should be changed or what should be done in a policies or with practitioners or with the scientists?
Yes, I've been saying this maybe before, but I think that this nitrogen fertilization should be maybe done like more often and in the nutrient poor sites as well, because now it's only done like once or twice at the end of the cycle. But this is really not a new idea. It's been already in the 80s when there's been like suggestions that maybe we should do this more often, because also these effects that Boris found, I believe that they are not like they don't show if you just do once an application, but when it's like repeatedly added, then there's more needles and more carbon and nitrogen on the soil layer also. But why it's not done is usually the money issue. So I would say maybe policymakers or decision makers could like think if we could support this kind of action. But also we should study if there's and do more research of this slow soluble nitrogen fertilizers.
Anni,
So that would be
maybe
something I
yeah.
could.
There is a question related to that. Maybe if you could
Oh.
continue, I can read it. It's about is there any plans to test slow releasing fertilizers in Finland? And Johan is asking because they have in his PhD project evaluation of all the fertilization project in northern Sweden, where this kind of pelletized sewage sludge from biogas plants were used. So do you have any ideas on these slow releasing fertilizers? What is your view on that?
My view is that it's not very well researched, and I just I read some conversations that there's some kind of policies that are really like for like forbidden, like it's not possible to even study some of those in Finland at least. I know that in Sweden there is something going on, and I know that some researchers are studying that, but I don't know more about that. So
yeah,
that's
how about this? Johan is saying after biogas, so it's been sewage sludge as such is not allowed to be used here, I assume. But But if you take it from the biogas plants, then it's a kind of different. So is it also not allowed in our case of the biogas
I'm
plant?
sorry, I haven't really studied this subject, but there's been a conversation about like somehow to do this pelletized, like coated fertilization, so that it would like slowly giving the nitrogen on the soil instead of just like quickly. And of course there's the leaching problem. So if it would be like bit by bit, then it would probably like inhibit the leaching a bit.
Okay, good. Thanks, Anni. We'll come back to you soon. In meanwhile, we'll take one for Boris. Boris, if you are with us. So Andis from Latvia is having a follow-up. So is it correct that fertilized soils release about 10% of stored carbon in our trials? If soil carbon is 100 tons of carbon, is it like 10 tons of carbon is lost annually? Is this true, Boris?
So yeah, so basically, if that would be, then it's the case. Yeah, annually it's about 10%. So I think the case is that whether the soils are in equilibrium. So usually the release, when we have commercial forests, then the liter input, if it is 10%, then they release about 10%. Yeah, but in this, in our case, we have seen that in the one figure of those 3 years, there was some kind of increasing trend. So control plots there annually they were losing 13%, but I think this should be also taking into consideration also for nitrogen fertilized how much what is the liter input, and if this one is if it is about 10% or 13%, that doesn't mean that they will lose all. So maybe nobody's implying this that they will soon lose all the carbon, but I'm just giving a note that all this percentage needs to be considered also with liter input.
Yeah, so the input is replacing the losses.
Yeah.
Okay, very good. Thanks. Then we have maybe this one goes to Anni. We have 2 questions here, and please, you can add more questions from the audience. If you have anything, please go ahead and write it down. Okay, there was this implementation gap of fertilizers in forestry versus research that you have done. What are the specific gaps in nitrogen management in current forest practices? What are the pathways to bridge the implementation gaps based on the research? Maybe, Anni, you touched some, you mentioned money was mentioned, but what other issues, what do you see kind of barriers for implementation?
Yeah, I think that there is this kind of idea that it's not profitable to do this in nutrient poor sites. Like, for example, this Karstula site it's very dry and nutrient poor, and I've heard that in practical forestry nobody would do this like this. So so I would like to encourage forest owners to also think about climate change, not mitigation only, but adaptation, because this would be probably, it might be in the future that these kind of nutrient poor dry sites really need this, what is it, karikekerros in English?
Litter layer.
Yeah, litter fall, yes. Litter in the soil litter for the resilience against, for example, drought. How to what was it? What would be the pathway to bridge the inflammation gap? Well, I think Tapio has those like forestry. Aleksi, help me. What is it like?
Kind of extensive services,
recommendations.
Yeah.
Yes. And so maybe we could like, and also like if there would be some kind of organization who would support this kind of repeated nitrogen fertilization, bit by bit, if it would be like done more, maybe in that way we could see that it's actually beneficial. And then so maybe we need forest owners who really want to experiment with this kind of and start to do it like that. Maybe we can see the benefits more when the climate is changing.
Maybe, yeah. Okay, thanks. Then we have a follow-up for Anni, and then after that I have one from Gustavo to Boris. But for Anni, there's this: do you suggest any fertilizers application frequently using chemical or any other biological nitrogen fertilization means? What is your roadmap? I mean, this was also touched earlier, but how do you see that?
Yeah, it is a good question. The only thing that I can say for this question is that we need more research. So I'm sorry, I have very boring answer to this question. Yeah. So different options should be researched, but it's on my table. Like maybe in the future I'm able to answer that question better.
Yes, thanks. Then Boris, there's a question by Gustavo. Has the same dose been applied at each time? Have any differences in litter fall and biochemical aspects been analyzed, and have these been taken into account when defining the end dose per hectare? Thanks.
Yeah, thanks for this comment. And I think the approach was uniform. Probably there was like slight difference. I think applying urea, but I think they try to apply this 180 kilograms of nitrogen per hectare per decade, once per decade, like at the beginning of each decade. And okay, now we can see that there was small differences.
Here's the amount.
Yeah, when the stand was established in 60s, and then they were experimenting a little bit, but later they changed to ammonium nitrate all the time, this 180. So, but okay, the rest of the questions, like if the different impact of like nutrients in litter fall was analyzed, I think probably with some other studies, but like in our case for this respiration, we didn't have during these years any analysis of differences between different nutrient content of litter fall. I think this nitrogen in needles that was analyzed from some previous years' measurements. Anni, I think you know better about those nitrogen in needles. So yeah, we didn't take these differences into account. We didn't have that data. Yeah, but one question that one comment that I agree or answer that like it's that I think that when we upscale or try to like apply this knowledge to larger scale, it should be noted that this is like one stand and little bit limited to these conditions. So applying it needs more research. But we need to analyze also those other stands.
Thanks, Boris. And yeah, there's maybe there's a last there's two guest questions. Maybe this last one is related to the discussion. Maybe I could briefly. So this it's about the chemical composition of the needles. Have that been studied? And I think in 80s it was studied, and some of that data was shown in the paper by Anni. But so there's data from the needles about the element concentrations, but only part of that data was used in Anni's paper, and it's from 80s, before and after fertilization. And then there's a second last question here in the list is that have you considered the climatic consequences of nitrogen management? There appears to be implementation trilemma, solutions for nitrogen rationalization, trade-offs, and economic bottleneck. So Anni and Boris, who wants to go? This is about relates to the economics of nitrogen fertilization and trade-offs. So briefly, if you can touch on this.
I will give floor to Anni.
Oh, I thought you have some idea. Well, the only thing that I have in mind that I haven't still maybe talked more is the leaching part. Like, what is the like environmental risk with nitrogen? And that's why I was thinking like maybe to add smaller amounts and like bit by bit, maybe that would be helpful. And yes, economic bottleneck. Yes, we already maybe said something about this, and we know very well that is a dilemma, because it's also like if there's like multiple times adding that nitrogen on the side, it's like a lot of work. So that's why maybe this slowly soluble nitrogen would be a solution, but a provided solution maybe in the future. But maybe it should be talked more, discussed more, maybe also researched more. Maybe we know something in the future better.
Thanks, Anni. Maybe I'll take the chance to take the last.
I could also.
Last.
Okay.
So. Sorry, Boris, if you have.
Yeah, I thought that I will just give Anni to start, or I can complement a little bit. Okay, there is a cost also of analyzers, and there is environmental cost for emissions. But for example, the emissions related to producing analyzers were much less, like magnitudes less than the benefits. But I think to know better economic models, we should do optimization of the benefits from the biomass and reduced emissions, and then we could compare probably, what would be the most optimal way of fertilizing.
Good. I think we have covered all the questions from the chat. Maybe I'll take the final question as a chairperson here. Maybe it's for both of you, because Anni showed that you can boost really the tree growth once in a decade by doing the fertilization. So you had heavier needles, and then Boris, you were showing that the relative respiration in fertilized plots were reduced. So I'm just puzzled and the leaching was mentioned here. But where is all that nitrogen going? Where do we find it in these ecosystems? Do you have any idea on that? because we have been adding the nitrogen there a lot. Where is it going?
You have questions to us?
Yes.
Do you want to start or?
No, start, please.
Because I have in this paper what I didn't explain in my talk. It was there's like nitrogen use efficiency that we measured, and it's actually quite high. Let me find it. So it's 0.16 cubic meters of tree stem volume per added nitrogen kilogram. And that makes, if we only calculate tree carbon, let me find that number also. Well, it's around like 40 kilos. I can't find the exact number, but I'm just looking. Well, I can show you this screen if we still have like time.
I think time is up, but you are saying that it went to the trees. So your reply is that it went to the trees. The added
a lot of it went to the trees, but of course I believe that some part of it also leads somewhere else. And then from the trees it fall down to the ground, but also it's poor site, so maybe there was some other plants also growing.
Good.
But yeah, Boris, continue.
Maybe some of it went to the organic layer, because I think we found that the organic layer was doubling. Yeah,
I agree with Aleksi. So it went to the trees and probably most likely to.
Okay, thank you very much, Anni and Boris. This was very insightful discussion, and thanks for the audience for the nice questions. And I think we are ready to close our HoliSoils webinar on nitrogen. And I want to wish happy and enjoyable weekend for all of you.
Thank you, Aleksi, for sharing. Bye.
Bye-bye.
Thank you, everyone. Bye-bye.